Have a
We
good
wanted
one.
to we wanted to welcome everyone. We have a great meeting today. Alice and I will give some introductory remarks and then we'll kick off to the main session. So Alice, do you want to kick us off?
Yeah, do you want to share the slides, Jesse?
Yeah.
Alrighty. Okay, so a couple of things we wanted to mention today. I want to say the one of the probably biggest thing that has happened over the past month, but truly, this is a big moment that the field has been waiting for a long time. As most of ASCO had some kind of assessments and guidelines published on based on very old 90s technologies to perform quote unquote functional precision medicine or like say drug sensitivity testing ex vivo. Those were very negative. Again, not, I don't think they were wrong necessarily. It was not the time for the field to jump ahead. And those guidelines are now formally archived and fully inactive. So ASCO came out with a strong statement, kind of retiring these old rules. I think it was a nicely well-formulated statement whereby they're not recommending for or against, necessarily any type of functional precision medicine approach, but they're open to evaluating these technologies fairly on the basis of the new data. And obviously, again, this is a very different field that it was 30, 20, or even 10 years ago. And so this opening, I think it's truly a fundamental moment for our community. They are supporting advancing this type of technology to improve patient outcomes. And so I think what it means for us as a field is that this is truly the time to build the evidence, the infrastructure, the validation, the standards, the pathway to kind of map the pathway ahead. I'm sure this is going to come up in Jeff's and Keith's discussion as well, so I don't want to belabor the point too much, but we really wanted to make sure that that these big change is kind of on people's radar. Just so we can move
Thanks,
on.
Alice. Yeah, it's certainly an exciting time, and it's also a time to ensure that SFPM can play a catalytic role in rallying many stakeholders to achieve many of the tasks ahead of us. We have a great board of directors that provides consistent input, a great membership from all of you, great partnership, of course, with the NCI. We're also beginning to increase our connections to biopharma and biotechnology industries. So today we're announcing for the first four members of our new President's Strategic Advisory Board. The PSAB will be a six-member body that will parallel our board of directors and help to provide Alice and I and our board and all of our members strategic guidance as we develop next steps as an organization. 10x Genomics, Tahoe Therapeutics, GSK, and Tempus have agreed to participate in our advisory board. They're making an annual contribution to the society as well, and they're ensuring that their members of their organization are intellectually connected. We're in late stage discussions with two additional members, and we expect to announce the full advisory board in the months ahead. But we wanted to publicly thank the first four members. We'll be hearing a lot more about the advisory board going forward. We've also put out a call for catalytic supporters. These are smaller organizations, typically, and family foundations and others that are interested in supporting the SFPM and their mission with annual contributions. They're invited to seminars, they're invited to have free memberships, and we co-publicize with our catalytic supporters. Cancer Commons, Ex Vivo Labs, Elephas Biosciences, Imprimed, Kyan Technologies, and Christian and Manu Regenbrecht have agreed to be our six initial catalytic supporters. And we thank them. This is really going to help drive the mission of the SFPM forward. And if you represent an organization that's not currently on this list and you'd like to be counted amongst our supporters, please reach out and let us know. On the website, which I'd encourage everyone go to, you can consistently find a full view of the SFPM. Can you see the website here?
No, we're still on the presentation, Jesse. I think you may need to reshare. Yeah.
On the website here, you can see consistently updated what the society is working on, what are the upcoming seminars, like Hans Klaver's coming up this fall for members, as well as a full list of the supporters with their logos and more information about them. So the website will consistently grow and take on new information, and we think that's going to be a great one-stop shop for everyone here to understand what SFPM is working on. Another call, if you're not a paying member, we'd really appreciate your $100 a year membership dues. Clearly, where we're headed is going to require full engagement. It's going to require cash to organize workshops and stakeholder meetings and launch pilot projects. So we thank you to all of the members. And if you're not currently a member, we'll put the note in the link so you can pay your membership dues during the seminar today. Okay, that's enough of an intro. Did I forget anything, Alice, Jeff,
I think
others,
that's
before we
great.
jump in?
Stay tuned for the next, we have a few meetings we're organizing. We're working hard to continue to bring the community together. Again, the time is truly now. And so we want to continue capitalizing and engaging the stakeholders and get us all aligned. I think this is part of what we're doing today. Very grateful to Jeff and Keith that agreed to do this once again. As most of you know, there was a very important NCI organized and Tony Letai, of course, our former president, driven workshop now about a month ago, right? I think Jeff reminded all of us of how fast time goes. About a month ago in Washington, D.C. That was really a fantastic workshop. I think there was a lot of excitement coming out of that. We had close to 700 registrants, I believe, for that event, which was, organized by Jeff Tyner and Keith Flaherty. We had a fantastic discussion the week after at our SFPM seminar, and we're, again, due to the overwhelming interest, we certainly did not go through everybody's comments and points. We invited Jeff and Keith to discuss again, where the field is going, what did we learn from the symposium, and what are next steps. I don't want to belabor this too much. I think everyone knows Jeff and Keith, but just by mean of, quick introduction, Jeff Tyner is a professor at OHSU, where he directs also the Cancer Graduate Program and the Translational Oncology Program for the Knight Cancer Institute. Obviously, very interested in developing functional and omics, data set in heme malignancies. Keith Flaherty is the Director of Clinical Research at Mass Gen, and also a professor at Harvard, obviously very well known for his work developing some of the key, targeted therapeutics that are, work so great for patients today, as well as leading the NCI match trial. I'm just gonna, leave it at that. And, and maybe I will mention that Jeff has been the treasurer of the SFPM since its inception, right? So he's one of our board members. And I'll let you guys guide the discussion from here.
Okay, great. Thank you, Alice and Jesse. That was outstanding. Yeah, so I think, this is really a continuation of the discussion that we started a few weeks ago, a month ago. I'm going to just put one slide up. We're not, hopefully not going to spend a lot of time talking at people, but really hoping to have a discussion because I think we had some discussion a month ago, but not everyone had a chance to make their comments heard. So that's really the main purpose of this meeting. I'll just put this up here. I showed this last time. This was in the concluding remarks of the workshop. Some ideas of some directions forward, certainly not all inclusive, about various funding mechanisms, potential for centralized labs, engagement with regulatory bodies, et cetera, et cetera. You can read all the bullet points here. I can give a quick update about, it's only been a few weeks, but one of the outputs that we want to have from the workshop is a white paper, and we've just recently gotten the ball rolling on that. Had to square away a couple of questions about who could and couldn't be included in the white paper, and that's now done. The idea of this white paper is going to be that it will summarize the content of what was presented at the workshop, the key discussion points that happen, and we do have good notes from the workshop for that. But then importantly, it's also going to lay out what we think are key recommendations for important next steps for the field as a whole. And we envision this paper being organized roughly around the sessions of the workshop and those various topics. And important to note that these recommendations are going to be for the field as a whole. They're not going to be specific to any one entity. They're not going to be specific recommendations for the NCI, for instance. Some of them could end up being, activities that the NCI takes up, but certainly not all of them. And some of them may be activities that would be important for organizations like the SFPM to engage with. So we really envision this white paper being a summary of the workshop and a series of recommendations for what are the key next steps for the field as a whole. And so that process has started, and we hope to get it going quickly and get that put together in the near future. So, this would be a great opportunity for anyone who has ideas, encompassed within this slide or in addition to this slide of key next steps for the FPM field, to have your voices heard. And we'd love to hear any and every idea that people have. I don't know, Keith, if you wanted to add anything to that before we.
Yeah, no, I think no, well framed. I mean, the scope of this conversation and ultimately this white paper is completely broad, as you said, like really kind of like everything and anything that falls in the domain of what this community thinks would advance functional precision medicine. And I think just to remind everyone that Tony was very clear with us, and I think we hopefully were clear with everyone who participated in the workshop, that his near-term focus is entirely on clinical adoption of functional methods, leaving aside the discovery slash sort of, let's call it discovery, but sort of the insights or the benefits to the field that could come from adoption of functional methods. But really focusing on assignment of patients to therapy as the ultimate goal, and using these methods for that purpose clinically. So he was kind of very clear that that's what he was after in terms of the workshop. And then similarly, I think the outcome of the workshop in terms of what the NCI is considering support of, both intramural and extramural activities, as alluded to here on this slide, falls into that frame as well. So in the conversation we had a couple weeks ago, and I think where probably we should maybe continue to focus a bit is really kind of continuing to collate, if you will, collect, discuss comments from this community regarding what they think would be kind of the most impactful NCI initiative. So again, Jeff's absolutely right that like there's going to be lots of other activities that are going to need to happen to lead even to Tony's goal of more widespread clinical adoption. And I think it makes a lot of sense to be thinking about what can the NCI do, and then at some point, even just from a fiscal perspective, there's going to be a limit to what the NCI can support, and so therefore there's going to be quite naturally then a larger scope, and I think still a need to coordinate with other nonprofits and of course for-profit entities as well in terms of trying to advance this ultimate goal. So I think Jeff and I have heard lots both at the workshop, maybe even before and after, on this topic, but I think that's kind of our purpose in the last convening of this group as well as this one, is to really kind of get people's view. And I'll just maybe kind of finish with kind of repetition of a couple comments that I made the last time we met, which is I think that the opportunity exists to envision some kind of multi-center clinical trial that would hopefully tap NCI resources, their ability to act as a regulatory sponsor of multi-center studies, but basically to frame some kind of idealized view of what would be kind of the dream scenario, and then maybe within that even starting to think through kind of like rank order priorities. So if you were to say, well, let's apply functional precision approaches to every single cancer type and every single therapeutic modality, great. Awesome to have that overarching framework. The reality is probably there's going to need to be some strategic focus within that. And so for people in this community to really have a voice in rank ordering what they think is really kind of low-hanging fruit for an intersection of sort of cancer types and therapeutic modalities, obviously through the lens of specific assays that are purpose-built to be able to choose amongst those and setting up a clinical trial anyway, assign patients to those therapies.
Absolutely. So I'm going to stop sharing at this point. I can always put it back up if people want to see it again. But I think at this point, we really like to turn it over to the floor and love to hear ideas, comments, questions that anyone has around these topics. I think there's already some stuff in the chat.
Jeff, do you want to call on folks or do you want us to MC? What would be the easiest?
Either way, it looks like there's already some comments in the chat.
I think there are a couple of quote unquote technical comments about the workshop and the products. I think you mentioned that there's been some discussion about who can contribute directly to the paper. So if you can clarify that, and also about sharing the decks, potentially also the NCI, I assume, materials and recordings, if you guys can comment on that.
So I think yes, in terms of the recording of the workshop, I don't know that it's yet been made public, but that is absolutely the plan. I think there's some steps that have to happen at the NCI in order for that to be released, but that's absolutely the plan. It will be made public and it will be available for everyone's consumption in the near future.
Jeff, this is Anna
Yes.
from
Yes,
NCI.
Anna.
So yeah, the recording is just about ready to be released, and there will be a message going to all registrants that the link is available, and it will be a link to a publicly accessible space, which means that people who were not registrants who go to that website will still be able to see it. If that makes sense.
Great.
All right.
Outstanding. Yeah, thank you, Anna. That's great.
Thank you, Anna.
Will this deck be available? I think we're happy to, yes, I think we're happy to send out the slide deck that summarizes the agenda of the workshop and these initial bullet points for directions forward. We could send that out certainly to SFPM membership or to the people on this call. And then I think, in terms of at this point, the white paper, we're really hoping to enlist the effort and the help of all of the panelists. So the folks who contributed to the workshop as panelists and chairs, and they have a great handle on the content that was presented, and we have good notes from the discussion. So that's the initial idea, is that this will be the group that will formulate the white paper.
Jeff, I'm glad you clarified early on that the audience for the white paper isn't exclusively the NCI, but is the community as a whole. As folks know, many of you have also contributed to giving the SFPM advice on where could this neutral sort of nonprofit society add value, and we're intercalating that advice into our own series of strategic documents. I think we're trying to be deferential, Jeff, to your and Keith's process. We want to let this vision paper come out first, but presumably it would give clarity to where we could add value, and dividing up the contents would be wise to do. I think there's a lot that the NCI won't, as you said, be in great position to do, and maybe SFPM won't be in great position to do either. But identifying that gap, I think, will help funders and other organizations, identify their place. Several of you have reached out to Alice and me recently and said, I'd love to do this. Can I join a work group to do that? And I think the pieces are still, kind of coming together in the next month. So I think there's going to be a lot for everyone to do together.
And
absolutely.
yeah. Tim, I know you.
I would like to make a suggestion. Maybe the leadership of this society can just make an outline, like a relay, so for example, if you write a paper, you have your section title, there's a sketchy plan. And so people say oh, I can contribute advice to this category, relay, how to increase interaction with the industry, or, something. There is a kind of a rough roadmap, or with some section titles, and then people can contribute.
Lynn, I think the SFPM is probably about 6 weeks away from having a pretty good sketch of a roadmap. And I think Jeff and Keith, I'm not sure what your timeline is for the outline of the white paper, but perhaps by mid-September, end of September, we can look at these two documents and answer Lynn's question of kind of what fits there. Is that kind of the timeline you guys are thinking about?
Yes, I think certainly not any longer than that would be my hope.
Like Lynn, I think there's a whole discussion about benchmarks and standards and metadata and ontology that probably are going to be easier for a neutral international nonprofit to take on than a governmental body. And then there's going to be other things requiring large amounts of capital to run clinical trials that are going to be ideally better for the NCI, to sort of take on. So I think you're calling for that mapping, and we agree.
Yeah, it sounds like, some of we all have a different expertise, even we have this common interest. So we want to find where we can contribute. And so if there's some kind of a map, and then we can say, hey, I can help out with this part, and then just contribute my expertise and my knowledge. So I think it's just more directional rather than just like you have 100 of a message coming in.
Yeah,
you don't
absolutely. And we intend for this to be directional, and I think that's the role of the SFPM, is going to be guiding some of this conversation. I do want to get to a comment in the chat from Venera, which I think speaks to the point that Keith was trying to make in terms of prioritizing, like who's going to decide, in terms of the clinical trial, even an NCI say, sponsored trial, what the priority is, right? Who's gonna set that conversation, and what are the cancers that you think people are going to be more keen to start focusing on?
Yeah, I mean, I think this is a really important question, and I think there obviously is going to have to be a process to evaluate different candidates, and I don't that will be up to some sort of review body that the NCI puts together, that will evaluate different tumor types and different assay platforms to make decisions on prioritizing. I think the criteria, there are going to be a number of criteria that will play into that decision-making process. One would certainly be tumor types for which there is, obvious room for improvement. That's going to be, where there's an unmet need, is going to be important. But also, tumor types for which there's a demonstrated platforms with a track record of successly performing tests and delivering results that are actionable. So I think there's going to be a series of different criteria based on the clinical unmet need and the sort of ready for prime time status of testing platforms in those tumor types that are going to be factored into deciding on how to prioritize the first tumor types that will play into a clinical trial.
Keith, I know you had that nice umbrella diagram, that one slide that showed kind of your thinking about how the pieces could fit together. Has your thinking kind of stayed in that place of multiple tumor types, multiple technologies, multiple kind of sub goals?
Yeah. Well, I think only for the purpose of trying to frame the largest possible scope in a way, right? I mean, just to remind in other words, there's nothing that's been predetermined in terms of thinking through like solid tumors versus hematologic cancers, or again, therapeutic modalities, and then certainly apropos to many people's work in this group, choosing an assay. In other words, if there were an NCI sponsored trial and or assays plural, that's the an important, I think, related point. So it's literally every variable is not yet set. And I think maybe one point worth just repeating from what Tony said at the workshop publicly is that he does not have a view yet about what entity within the NCI will be the decider / framer of a study that the NCI might be the sponsor of. And that's an important point, because I mean, he's actually, even at the workshop, he said this. He's said it before. I remember at the ACR annual meeting as well, in a sort of fireside chat, that he's not sure that the cooperative groups, for example, are the ideal construct to think about, and that maybe a more bespoke consortium might be best here in terms of even just thinking about the clinical sites. So what that means is that with Jim Jorshow as the head of DCTD, which is the largest relevant umbrella within the NCI, within the scope, for sure, this initiative will fit. That Jim and Tony and other leadership at NCI will determine kind of how to convene a set of decision makers from within the NCI, and then perhaps inviting outsiders. But again, nothing's been specified on that topic. But I guess everyone should make no assumptions about the idea that that there's going to be a concept that's going to go through "quote unquote" conventional channels like the cooperative groups, for example, or ETCTN with the Early Therapeutics Clinical Trial Network.
Tim, I can see you making a number of comments in the chat. You're certainly welcome to share them. I think your comments are centered around kind of assay development and readiness and CLIAization, if that's a word. It's a super important topic and an expensive and complicated topic. And it's one that Alice and I have heard in our stakeholder interviews quite a bit. Can organizations share information about CLIAization? Can the SFPM or other potential funders or partners help to offset some of the costs of CLIAization? Could the NCI initiative do such a thing? I think it's a super important question, and we don't have the answers to that. I'd love to wake up in a world in which there are 17 FPM clinical labs across the country in a diversity of settings that future initiatives like this could draw from. I know, Cliff, you're thinking about this, too. And it's kind of unclear whether the NCI, its mechanism itself, would accelerate that, but maybe the SFPM can help as well. I don't know, Alice, you've thought about this a lot as you've set up your own CLIA lab. Do you have any comments on that?
Yeah, no, I mean, it's obviously an important topic. I think kind of tying into what came out of the workshop, and Keith, correct me if I'm wrong, is that to some extent, the goal of Tony, as again, both Jeff and Keith stated, is to get things through to the clinic quickly. Some of it means that, the assays which are closer, I guess, on the regulatory path to that, accreditation or through to that accreditation are probably better positioned to have a quicker impact. That doesn't mean that is the only assay. And I think, just as Jesse said, as a community, there is space here to build, develop potentially resources to help, folks see assays through. Getting an assay through a clear cap process is not easy for an academic. And maybe that's a question also for Keith, as you guys, kind of think about this potential clinical trial. Like, would there be an opportunity for, say, a smaller company that has a clinical assay to be a part of that conversation?
I mean, I would say prior precedent would suggest yes. Like just going back to NCI Match, and then the sort of derivative trial, second gen match trials that got launched thereafter. In both of those contexts, there was an RFP, essentially put out by NCI/Frederick, open to private sector entities to come forward with diagnostic assays that would be used in that case, just for standard NGS/omic assays. So it's those experiences would suggest that that's probably what the NCI would do again here as well. I mean, in other words, open to certainly academically derived and validated assays, but also private sector ones as well.
Yeah, I do have something to say. As a pathologist, most of the CLIA labs are under the pathology department. And I've been like a pathologist running NGS, not NGS, it's just like we call it a molecular lab. And starting from PCR, FISH, RT-PCR, so forth and so on, like moving into the next-gen sequencing area, doing both DNA and RNA. So that's my background, and I'm very familiar with CLIA and how to maintain the CLIA status and throughout years. So I have 20-something years under my belt. And then it's really, this is a good starting point. So the functional precision medicine rely on assay. You cannot talk about the functional precision medicine without assay. But the CLIA assay, the key part is really the reproducibility. I mean, like really, and then I also worked for a short period of time on the industry side. And then we realized, really during that experience, many academic papers cannot be reproduced in other labs. So for the clinical lab, and then, the NGS quickly beat AML, that was a successful story Tim also mentioned. Because NGS is easier to do, the procedure is easier to standardize, and then industry really Illumina and the other big company put out all these standardized tests. And then for the clinical laboratory, you open the box. You they have end-to-end solution. You have to make it so easy, and so turnaround time is always an issue. The reproducibility is an issue. So if you if we can prioritize, and we have responsibility to the public, and then if we can pick a few highly reproducible assay, and then for particular cancer like really AML, CLL, my favorite, and then it's just really just put out a few stories, and then with a very robust, reproducible, clear assay, and then that will get us started into
Lynn, I think
into clinical practice.
Yeah,
I think. Go ahead. Go ahead, Jeff.
Yeah, I mean, I would echo much of that. I mean, I will say when we CLIA certified our assay 15 plus years ago, we looped, we did it under the umbrella of the Path Lab at OHSU. So anyone's at academic center, go to your pathologist if you want to CLIA certify your assay, because they can be immensely helpful. But the other thing I would echo is that, yes, I mean, as we think through, as whatever the review body is that thinks through how to prioritize which tumor types, which assay types, it's going to be in everyone's best interest to make sure that the first few platforms and tumor types that go through the process are successful. And so we really want to ensure that we choose carefully and choose tumor types and platforms that can deliver successful results. Looks like Pam has her hand up.
Thank you. So I just wanted to I appreciate recent comments, but the process is so different. It's so complicated that for these assays that so I was the medical director for a CLIA test since 2014 in Seattle, and I'm now the laboratory director. And I think these assays are so complicated, and trying to obtain the validation for these tests is extraordinarily difficult. We don't have our tests. Part of it stems from the fact that we don't have an easy way to reproduce those assays in other laboratories and have them be identical. And then we're trying to get increasingly more complex assays to be the best assay. So all that discussion that took place a month ago, described assays that can only be done at the one place where they do them. So for example, the chemo ID assay, it sounds like they don't believe that can be just done anywhere. So I think that the assays that we think look the best or are the best are not straightforward. And so the CLIA inspectors they're asking for proficiency testing, and they're asking for approved clinical proficiency testing. And these assays are never going to be able to conform, or there is no test, like you're supposed to do the test, and there is no test that you can do to prove it. So it makes the process extremely difficult. And I recently just went through 4 months of trying to get through all the current requirements after all these years, a dozen years. And so I would say that the idea that everyone will have that we'll have CLIA tests, that we'll be able to do our best assays, our most complicated assays will be CLIA approved instantaneously or quickly. I think that the people who have CLIA labs, like I think I'm in a good position to be able to add another test or to do another test or to use everything that we've got and perhaps bring that to a different laboratory. I think I'm in a better position, but I think it's naive to think that we can that we can just invent these functional assays instantaneously and get them approved within short periods of time.
It's definitely going to take a period of time. I want to make sure we get everyone involved here. I think there's going to have to be a working group. Maybe Pam, you and Lynn and others can be parts of that to support other labs across the country and around the world who are kind of thinking about these questions.
Yeah.
We've talked a little bit about how there should not be standards because standards kind of can get in the way of innovation, but there might be kind of a minimal information about kind of equivalent for an FPM assay that we could help researchers who are thinking about assessing reproducibility or quality control or whatever to internally validate and evaluate their assay against a set of metadata that this field believes are important to credential a particular assay. So I think that type of thinking could be a value add for members of the SFPM community and the peer-to-peer support. I'm thinking about a CLIA certification process. What should I do? Where should I start? What should I avoid? State, federal, international, like the comments are coming in. I think Pam, you and Lynn and others could be very helpful here. I think just supporting members through that process could be helpful.
Yeah. Years ago, I actually traveled all the way to France to try to help them set up their laboratory. I mean, I've been trying to help people for a long time, but I think that the process, the inspection process and the requirements have massively escalated. The current cap, the cap requirements are quite onerous now, and I think that because of more recent changes, I think it makes it even more difficult than it was. So this idea, the only reason I'm mentioning all this is because it seemed like Tony wants it here and now, and I think it will work for the labs that already exist, and maybe we can just try to get another assay approved in those labs that already exist. I think it might be I used to think it would take maybe 6 months to get the CLIA, but I think it's going to be longer.
But two big solutions. Okay. Like, so Pam, I, through my 25 years experience, and then one is to have a commercialized assay. So you have a diagnostic company working on the assay. They sell a box of reagent. The technologist was following instructions like assemble IKEA piece of furniture, 1, 2, 3, 4, 5 steps. And then those help like tremendously in the case of doing BCR able to monitor CML, their response to therapy. This is I went through the entire process.
Yeah,
I think we need to move on. We need to move on to some other topics, but I think we've heard the point loud and clear that this is a very important.
The second solution is a central lab. I'm just pointing out what solution 1, commercial lab. Solution 2, central lab.
Yeah, that could be very helpful.
Like to encourage the community to keep in mind that this is a very diverse ecosystem. Not everyone is at the same place. Many people have seen things through regulatory landscape. I also would like to maybe push the community to think that the part of the reason why it's so challenging to see these very high complexity assays through the CLIA/CAP process is that the CLIA/CAP process is not built for this type of assays, and so we are always trying to squeeze and kind of push and pull something that is round into a very square box, and that is very challenging. But this is why the NCI making this push and us as a community making this push are making things change, right? Like ASCO had not made any changing their determination for the past, whatever, 20 years, and now it's changing. And so I'm not saying that that process is going to change tomorrow, but I think we do have an opportunity now as a community to make our voice heard. That again, I think, as someone who is seeing a clinical lab, an academic lab right now through ClearCap, literally, I can tell you, it's not undoable. It's very onerous. The resources that need to be put there are challenging. And again, most of it is because the rules don't fit the assay. But I don't know, maybe Keith or Jeff have some, like maybe there is a place there to start those conversations and encourage those organizations as well to think through where the world is going.
Yeah.
Melissa, I just want to make a comment. Can you hear me? Related to what was brought up at the NCI. I think what many of us are trying to get ClearCap, we have a ClearCap commercial lab, but even with that, what I'm trying to do, and it's been 2 years, is to generate all that data that we need and publish it. I think what we need to do here as a society is try to help each other by publishing all the validations that need to be done to get our assays ClearCap. Yeah. So even though it's been 2 years and we've generated 20,000 data points, I think what I'm trying to do now is to at least put things together so we can publish it, and that really helps everybody else understand what it takes to make a ClearCap lab or get this assay validated.
They're supporting individual publications, and then we heard from our colleagues at Friends of Cancer Research about their efforts to take, let's say, a shared set of 100 samples, maybe organoids in this case or something, send them out to 20 labs and actually have a community project for everyone to run the same samples against the same assays so that we all can understand together where are there similarities and where are there differences. I could see a community challenge project like that being something that SFPM and Friends and others could run. And I think that would help us understand a lot what signal to noise level is actually needed for the type of reproducibility that we're kind of thinking about, and how does that vary assay to assay.
Yeah,
Can we switch topics? Oh, sorry.
I just would super quickly underscore your mention of Friends, because I think Friends would be an ideal partner for this society to think about that CapCLIA
exactly
reform initiative like that. The NCI is unlikely, almost certainly not going to be the entity that kind of tries to approach CAP in terms of negotiating such.
Europe has many regional or central labs already for diagnosis. Yeah, through the BCR-ABL experience, I actually got to talk to many more really European colleagues.
Let's move away from it. Let's move away from
overall.
Disrespectfully,
the US is more chaotic.
Yeah, we have 15 minutes left. Let's move away from assays, and there's some comments around patient advocates and their roles. Keith, I also want to come back to drugs. Which drugs? We have some pharma people here on the call. Like, what types of pharma partnerships and what types of advocacy partnerships do the two of you and do we all kind of think are going to be needed? I don't know if you have comments on those.
Well, I'll start with drugs since that's my favorite topic. What the point I was trying to make in the workshop and since is, I think this community knows best what assays deployed in what tumor types, can discriminate amongst certain therapeutics best, right? And I would just underscore that word discrimination, right? So this is it's a huge challenge to try to demonstrate the power of functional methods if you're working with 10 therapeutics that all fall within one really tight class. I mean, it's just to be extreme about it. Like if you had a bunch of MEK inhibitors and a bunch of ERK inhibitors, good luck, right? I'm not saying that there wouldn't be discrimination, but it would be probably pretty narrow. So that's where I think, based on this group's experience, really trying to think about, kind of a rank order of, okay, well, what are those indications where we already have sort of, evidence-based approvals and use of therapies, and then look across the set of those therapies, and you'd say, well, there's a bunch of different modalities, and we've already got assay evidence that you can really cleanly discriminate, like non-overlapping Venn diagram of sensitivity to those therapies alone or plausibly in combination. To me, that's a really critical piece of this exercise in terms of trying to provide the best advice to NCI about where to focus and where there are areas where the therapies that one could envision deploying can be best discriminated. And just to be complete, That could include investigational therapies, of course, right? I mean, this is obviously when thinking about an NCI sponsored trial. I think one should be considering anything that's sort of post phase 1 as being potentially within bounds. And our experience in MATCH and the sort of second gen studies that came out following MATCH is that biotech and pharma were just unbelievably supportive in terms of making their drugs available for those studies when asked. And we had processes for vetting, what we thought were the best assets against, any given target and so on. And, I can't promise, on behalf of pharma and biotech companies that will, always be true. But I would kind of hold aside concerns, about that, and really just again focus on what we think is the kind of, dream scenario of a collision of cancer type therapeutics and assay. For the purposes of really being able to parse out, kind of patient treatment recommendation in the context of a trial, where one can then anticipate that you'll really be able to show the largest possible benefit versus, either just as I've suggested before in single arm phase 2 mode, where you're just demonstrating that you can actually match patients to therapy to which they get an unquestionable benefit, aka durable response, or if conversation continues to migrate towards randomization, fine. That's not Tony's mandate, at least at the outset.
Jeff, do you?
Yeah, I mean, I completely agree. I think that, I think we need to be realistic with with our considerations about platforms and drugs. And I would say that, most of these platforms, the ones that I'm familiar with at least, they perform really well on certain classes of drugs, and there are other classes of drugs that they don't perform as well with. And I think, we can always work to improve that and try to capture all drugs at some point. But I think with our initial stabs, we need to be realistic with any given platform about which drugs are predicted well and which really don't perform well in the assay. And that needs to be a really big part of the consideration of the tumor types and the platforms and the drugs that are going to be evaluated in any of these initial clinical trials.
Yeah, the targeted therapy are fundamentally so different from immunotherapy. And then this is perhaps will require different type of assays. We already heard, so many talks on so the strengths of each assay for a particular therapy, I can't agree more.
Yeah, I mean, certainly immunotherapies versus small molecules are two very different entities, but even within small molecules, there are different classes of drugs that have different kinetics, that have different phenotypic endpoints, and depending on the platform, the certain classes of drugs will or won't be predicted as well based on the platform and how those drugs work. So I think that's going to have to be an important part of the discussion and part of the review process.
And I think thinking about this strategically, I don't know if Dave Tuveson's on the call, but to be a bit provocative, Dave said, what you guys should do? Nail the KRAS therapeutics combinations problem. If you could do nothing else but do that and show that these assays are valuable, that would galvanize the whole field and showcase the potential. That's a bit of a different, I'm kind of neutral. That's a bit of a different construct than take 1,000 patients and select which of 100 therapeutics or 20 therapeutics are going to be best for those patients. Those would have different types of impact for different communities, but we should kind of struggle with those two extremes and figure out, what's in the middle. Levi said something important in the room that had stuck with me. He sort of said, do we yet have examples like BRAF mutations predict BRAF, inhibitors, of course, but are there examples in the absence of a BRAF mutation where the FPM assay predicts a similar strength of response that couldn't have been predicted from genomics, and then you have seen that the patient does respond? I think those types of extraordinary responses with, again, extreme signal to noise that can't be predicted easily from genomics, I think will help to turn a lot of heads. And I know we have anecdotes of those across the community, but if the future platform or set of approaches could increase the chances of finding 27 extraordinary responses through FPM assays, I in particular think that would be very valuable, as opposed to lots of predictions of 10% improvements. I personally think that might be less influential. I don't know, Keith, if you agree or disagree with that.
Yes,
sentence.
No, that's the point I've been trying to make about the kind of focusing on the idea of are there combined settings, cancer types therapies assay where you could, reliably score a, again, durable response rate that is just, above and beyond what can be achieved. I mean, Tony, again, just to remind people, was grounding the conversation initially on patients with relapse refractory cancers, which doesn't mean eighth line, but like have failed the therapies that, like, unquestionably are beneficial, as in improve overall survival with acceptable toxicity. That's usually one line of therapy per any given cancer type. And so he's really wanting to focus on kind of an early relapse refractory discussion. That's not the only place that we could be thinking. I mean, both in terms of advising, NCI on most impactful context, but again, if you've got that, if you stick with that framework of patients who failed like the best available therapy, you can certainly say that it's not possible with any other approach to come up with a 50+% durable response rate, or just pick whatever threshold number you want. But like, I just, that was my urging in the last conversation to, think about, kind of what is just, a level of impact, even amongst a relatively small number of patients, that would be really hard to argue against.
Yeah, I mean, it seems like there's, at least three different major buckets here. There would be, one approach that would be to take, a tumor type for which there are at least two or more frontline or second line therapies for which genomic and clinical features alone do not give us certainty as to which patients will benefit from which of those regimens, and for which we think that an FPM assay could improve the predictivity of patients who will benefit from those standard regimens. That would be one approach. Another would be to go after novel combinations that are added onto the backbone of an approved agent like KRAS inhibitors. I'm, of course, partial to venetoclax myself, but there's a variety of options for that. And then a third, Jesse has mentioned, would be sort of repurposing of targeted agents in a marker wild type population, and illustrating that marker wild type patients can benefit from those targeted agents as well. Those are three very different formats, all of which could be interesting.
Cliff, you're trying to get in, and then in the last 3 minutes, I want to challenge people who have said nothing and have not contributed yet to have space to register their comments. Cliff.
All right, I'm achieving that goal that Jesse just gave
You've
me.
already commented, but it's okay.
All right. So there are a lot of people on this call who now, with the kind of the brave new world of the Tony Latai leadership and the interest in functional precision medicine, would love to write a grant proposal to NCI. And though recognize, and the conversation came up briefly at the workshop, that the study section, Tony doesn't control that anymore. It's back at NIH. So Keith or Jesse or Jeff or anyone, do you guys have any advice about how to go about, rather than just waiting for an RFP to come out? How do you go about presenting a fundable plan to NCIs in such a way it doesn't get read by people who say, it's another functional precision medicine garbage that'll never work?
It's probably more than a 2-minute discussion. I'm guessing there's no easy answers to that, Cliff, but if people are successful in getting certain grants funded, perhaps the SFPM can help them share, successful applications or excerpts from successful applications with their peers. So maybe Alice, we can bring that back.
And let me put it to Keith. You've talked to Tony about this more than any of us, I think. Any thoughts on that matter?
He has said publicly that the way that review works now is, setting aside panel constitution, is that obviously scores are given, but that pay line is no longer kind of the rubric by which decisions are made, but rather that like senior leaders, including Tony, see all grants and ultimately decide kind of what to pull across the line in terms of funding. So you just have to assume that, I mean, this is not the only priority the NCI has currently, but it's obviously a stated priority. So I think until there are like sort of dedicated review panels, as Jeff has suggested, could be envisioned. Till that day is realized, I think one has to hope and presume that even without a formal RFP, to be able to put in your application that there's going to be a supportive tailwind. But I totally agree with you, Jesse, that kind of sharing experiences around review process, for successful applicants, I think would be useful.
Yeah, when the EGFR papers came out about genomics and the value for lung cancer, all of a sudden review panel said, "Oh, genomics is useful."
Yeah.
So I think like really impressive papers that demonstrate the value that will go a long way, Cliff, to helping. And I know we can't manufacture that overnight, but whatever we can put in motion to enhance that. Of course, you're the first one to argue that focusing on non-governmental funders is also important. So if the SFPM and other entities can come together and help to build funders coalitions or organize white papers and have stakeholder discussions, I think that could be very valuable. We haven't finalized plans for how to do that. In the final minute, has anyone would like to comment that has not yet commented? Or Alice, you clearly have.
Maybe we can say a couple of things first. Anna very helpfully actually pasted the links to a couple of funding opportunities in the chat. So we will, share these links also in our next email with the community. There are opportunities out there already from the NCI that may be fitting. We cannot control the review panels, but at least, there are parts that may be, meaningful there. The other thing is I've seen quite a few comments in the chat about interacting also with European agencies and, European partners on that. And this is something that the NCI obviously cannot do. We have discussed this last time as well, but it's definitely something that we as the SFPM are engaged with. We are international by definition. So definitely, interacting with those partners is something that we are probably better positioned to do, of course, than the NCI, and happy to continue doing so. And then, Jesse, I think we're at time.
I think so. We did not have time for the patient advocacy discussion. We should certainly bring that one back. There's also some discussion in the chat about AI and AI models. I know that's important to all of us in many ways. And Alice, I think making sure we continue to have speakers around AI applications to FPM this fall, I think would be very valuable. Jeff and Keith, any final thoughts before we close it out?
I'll just say thank you for all this great input, and we look forward to moving this forward together as a community.
Hello, can you hear me? This is Chris.
Chris, any final thoughts?
Yeah, the patient advocate thing that you just touched on, I think that is a massively untapped resource that you guys are probably already aware of. But these people have enormous, obviously interest and organizational capacities and passion more than we do probably, and getting them like a third at the table is going to be huge. So you guys already know that, but we have several who are very, very, very articulate and really, connected and would love to participate in that effort.
Thanks, Chris.
Yes, you're welcome.
That's going to be very valuable. So it's going to be a busy fall. Jeff and Keith are going to get all of our brains to contribute to white paper. Hopefully, Alice and I can get all of your brains to help provide guidance for the SFPM. And there's going to be room for everyone to contribute in a number of ways. And I really do think this fall we'll have the answer to Lynn's question about who should do what and how can we all, drive forward together. But continue to contribute, continue to send ideas, and please continue to engage both with Jeff and Keith and Tony as well as with the SFPM. We'll see you in September. Have a great end of the summer, and make sure to take a little bit of time off and reflect on the future of FPM on the beach with your family.
As Tony invited us to do. Thank you, Jeff.
See you later.
Bye-bye.
Thank you all.
Cheers.